A seemingly healthy patient receives a standard genetic test with an alarming result. They carry a high-risk mutation for a severe metabolic disease. For years, the working rule in biology was that one gene equaled one disease. Under that reductionist logic, this patient's health was already a foregone conclusion. Yet, despite the results, the patient remains perfectly healthy. This outcome is a mystery only if we view the body as a collection of isolated parts, rather than an interconnected system. The human body operates like a biological symphony. No single instrument plays in a vacuum. Every note is influenced by the rest of the ensemble. To understand why this patient stayed well, we have to look past the static DNA and map the dynamic flow of data through the 6 omics. That flow of data begins right here, deep inside the nucleus of a single human cell, where the foundation is laid. This is genomics. It's the study of your entire inherited library. For our patient, this layer confirms the presence of that high-risk mutation. But the genome is just a blueprint. It provides instructions, but it doesn't have the power to execute them or cause a disease on its own. For that, we look to epigenomics. This layer acts as the body's software, using chemical tags that sit on top of the DNA to dictate which genes are active. In this patient, a healthy lifestyle prompted the epigenome to act like a dimmer switch, physically locking the mutated gene into an off position. Having a dangerous genetic blueprint matters very little if your lifestyle triggers the epigenome to keep those specific sequences silenced. Once a gene is silenced, the rest of the cell needs to receive that message. This is transcriptomics. It's the study of RNA messengers, temporary copies of active genes that carry instructions out to the rest of the cell. Looking at the patient's transcriptomic data, we see normal RNA being produced, while the level of mutated RNA is non-existent. The disease message was never sent. However, messengers are only words. To see the actual work being done, we have to look at the cellular machinery. That brings us to proteomics. This field studies proteins, the complex physical structures that build your tissues and drive every chemical reaction in your body. Because the patient's mutated gene was silenced at the messenger level, their proteomic profile shows perfectly functional machinery, with none of the broken gears that a mutation would typically create. Following the evidence from a dimmed gene to these healthy proteins, we can see exactly why a theoretical genetic risk never turned into physical damage. As these protein machines work, they produce and consume small molecules, leaving behind a real-time chemical trail of their activity. We track this through metabolomics. The metabolome is a collection of small molecules like sugars and lipids, providing a snapshot of your physiology right now. The patient's metabolomic profile confirms their daily energy production and nutrient processing are optimal, proving they are healthy right now. But human health isn't just about human cells. We have to account for the massive non-human variable living inside us. This is microbiomics. Trillions of microbes in the gut interact with our cells to regulate metabolism and train the immune system. Since these microbes influence how we absorb nutrients and respond to inflammation, a medical profile ignoring them misses a crucial system determining whether a disease takes root or stays dormant. If we had relied only on a genomic test, this patient might have faced aggressive, unnecessary treatments for a disease they were never actually developing. By investigating all 6 layers, we see the full story. How lifestyle, gene regulation, and the microbiome collaborated to keep the patient healthy. This is multi-omics. By integrating these 6 data streams, we can move beyond guessing and run a unified, predictive simulation of a person's entire biological system. We are moving toward a model where we don't wait for a system to fail. We can now interpret your unique molecular symphony and fine-tune the performance while the music is still perfect.